I am Dr. Anjani Devi, Associate Professor, Department of Biotechnology, Vignan's Foundation for Science, Technology and Research. As you pretty well know that I am dealing with the course Enzyme Technology, and we are presently dealing with the Unit 3 of your Enzyme Technology. In the last sessions, we have dealt with the kinetics of single substrate reactions. We have already covered the estimation of Michaelis-Menten's parameters. Along with that, we have also learnt about what is the importance of your Michaelis-Menten parameters like Km and Vmax. Along with that, we have already studied about multi-substrate reaction mechanisms and kinetics, and we have focused upon the turnover number also. In the present sessions, we are going to study about the remaining topics of your Unit 3. Then, I'll start with the topic which is nothing but your types of inhibitions. What are the various types of inhibitions? What is an inhibitor? What the inhibitor is doing? Why it is not allowing the enzyme to combine with the substrate in order to give rise to enzyme substrate complex and thereby to the formation of your product. So, we will see what is the effect of inhibitors on enzyme substrate reactions in this present session. First, we need to know what is an enzyme and what is an inhibitor. So, what is an enzyme? As we pretty well know, as we have been studying in this enzyme technology, that enzymes are mostly proteinaceous in nature. They are involved in various biochemical reactions taking place inside our body. So, what is the main function of these enzymes? Enzymes are proteinaceous in nature and they are involved in increasing the rate of reaction without changing the equilibrium of the reaction. And very important property of enzyme is nothing but your specificity. And it is highly selective catalysts that greatly accelerate the rate of reaction. It will be increasing the rate of metabolic reactions. There are certain molecules which can inhibit the metabolic reactions. There are many sort of molecules which are capable of interfering with the activity of this individual enzymes, and all this, the molecules which act directly on the enzyme to lower the catalytic activity is nothing but your inhibitor. So, what are these inhibitors? What is the nature of these inhibitors? Some of the inhibitors are naturally available. So, some enzymes inhibitors are normal body metabolites. They are present inside our body only that inhibit a particular enzyme, while other inhibitors are foreign substances such as drugs or toxins. So, what is the range of inhibitors? It may be a naturally available metabolite inside our body, or it may be a foreign molecule such as toxins and drugs. The inhibitors may be a part of your normal metabolic control of the pathway, a disease condition, or either a therapeutic measure. That means there are both advantages and disadvantages with this particular inhibitors. Sometimes these inhibitors are involved in therapeutic measures. They are involved in treating certain diseases also. Sometimes they are involved in stopping, hindering certain important reactions. So, they are causing both advantages and disadvantages. If your inhibitor is acting like a therapeutic agent, then it is advantage for you. If it is controlling certain metabolic reactions which is very much needed for your body, then it's a negative aspect of your inhibitor. Thus, the effect of your enzyme inhibitors could be either therapeutic or it may be lethal also. Now, I'll explain you what is the various types of enzyme inhibitors. So, there are majorly three different types of inhibitors. First one is your reversible inhibitor, second one is your irreversible inhibitor, and the third one is your allosteric inhibitor. let me discuss with you the further classifications in this. In the reversible inhibitors, we have further three different types. One is your competitive inhibitor, second one is your non-competitive, and third one is your uncompetitive inhibitors. And we can study suicidal inhibitors under the category of irreversible inhibitors. So, I've already told you that a inhibitor may be a drug molecule, or it may be a natural metabolite that is produced inside our body. It may be a antibiotic, it may be a toxin, or it may be any other natural product also. And these inhibitors may be organic as well as inorganic in nature. So, we'll start with irreversible inhibitors now. What is irreversible inhibitors? What can be studied under irreversible inhibitors? As you pretty well know that in this irreversible inhibitors, the major characteristics of these irreversible inhibitors are they will be binding very strongly to the enzyme and they will be completely inactivating the enzyme. So they bind tightly to the enzyme and they inactivate the enzyme. This is a major important property of irreversible inhibitor. Along with this, the inhibitors which bind reversibly to the enzyme oftenly forms a covalent bond to an amino acid residue that is present in the active site of the enzyme, and they will be deactivating or inactivating the enzyme permanently. So what an inhibitor is doing? Inhibitor, just think that this is your enzyme molecule, and definitely you know that every enzyme will be having a site which is nothing but your active site, which is specific for binding to your substrate molecule. Now, what an inhibitor is doing? It is going and it is binding to the amino acids that are present in the active site. And what is the bond with which it is binding to the amino acid residues? It's nothing but your inhibitors are binding to the amino acid residues with covalent bonds, which are very strong bonds. Because of formation of these covalent bonds, there is a permanent change in the enzyme. The enzyme is getting inactivated permanently. What are the different amino acid residues which are mostly susceptible, which are mostly affected by these inhibitors or nothing but the serine and cysteine. These two amino acids are mostly affected by these inhibitors. So, these serine and cysteine are having the reactive groups which are nothing but your OH group and SH groups. So, now I'll give you some examples of inhibitors. So, I'm just considering an inhibitor which is nothing but your DIPF. What is the full form of this DIPF? It is nothing but a diisopropyl phosphate fluoridate. In short, we are calling it as DIPF. This is an inhibitor. What is the enzyme inhibited by this particular inhibitor? It's nothing but, just consider acetylcholine esterase. This is the enzyme which is mostly affected by your DIPF. How it is going to affect? So what it is doing is, this DIPF, it is nothing but it's a component of your nerve gas. It is a component of your nerve gas. What it is doing, it is just going and it is binding with the serine residues that is present in the active site of enzyme, which is nothing but your acetylcholine esterase. Because of this, the transmission of nerve impulse is getting stopped. So, because of binding of this particular inhibitor to the active site of this enzyme, that is nothing but your acetylcholine esterase, your neural transmission impulse, nerve impulse transmission is getting stopped. I'll explain you with another example. So, let us see this iodoacetamide. This is also another inhibitor. So, this iodoacetamide is affecting the cysteine residues present in the enzymes. It is modifying the cysteine residues. So, your iodoacetamide have a property of modifying the cysteine residues present in the active site of various enzymes, because of which it is utilized or it is used as a diagnostic tool in determining whether one or more cysteine residues are required for enzyme activity or not. So, as this particular iodoacetamide is going to modify your cysteine residues, it is used as a diagnostic tool to determine whether one or more cysteine residues are responsible for your enzyme activity or not. Let's go for another example. Just consider an antibiotic penicillin. We know about penicillin. It is produced from a fungi and most of the people are very much susceptible to your penicillin. So, just consider this antibiotic penicillin. This is your inhibitor now. And what is the enzyme which is getting infected with this particular inhibitor, which is nothing but your glycopeptide transpeptidase. Just consider the enzyme glycopeptide transpeptidase, which is getting affected by your inhibitor penicillin. How penicillin is affecting this particular enzyme? Generally, this enzyme is involved in forming cross linkages between NAG and NAM units present in the cell wall. So, because of this particular penicillin, there is hampering or there is no proper formation of your cross linkages. So, what is a non-specific method of inhibition? This is nothing but your physical or chemical changes which ultimately denature your protein portion of your enzyme and are therefore it is hampered or inactivated irreversibly. So, because of this non-specific methods, your protein is going to get totally inactive. So, under the heading irreversible inhibitors, we are going to study about this suicide inhibitors. What are these suicide inhibitors? I'll explain right now. A special type of irreversible inhibitors, as we have told you that the suicide inhibitors are also binding irreversibly or inactivating the enzyme irreversibly. That means completely the activity of your enzyme is lost. And then relatively unreactive until they bind to the active site of an enzyme. So these inhibitors will be activated only when they will be binding to the active site of an enzyme. Until then, they are not in an active mode. They will be only getting activated, they will be gaining their reactivity once they will be binding to the active site of an enzyme. Moreover, they use the normal enzyme reaction mechanisms to inactivate your enzyme. They will be using the normal enzyme reaction mechanisms and we are calling it as mechanism based inhibitors or transition state analogs, as these inhibitors are using the normal reaction mechanisms to inhibit your enzyme. We are calling them as mechanism based inhibitors or transition state analogs. Besides, this suicide inhibitors that exploit the transition state stabilizing effect of this particular enzyme results in the higher enzyme binding affinity than do substrate based inhibitor designs. And designing a drug that specifically mimics the transition state is really very difficult because your transition state is highly unstable state and it is poor characterized structure is available in the transition state. So, designing a drug for a transition state is very difficult because of its instability. Besides, this suicide inhibitors, they remove the enzymes and they reduces the formation of enzyme substrate complex. I've told you that they won't allow the substrate molecule to go and bind to the enzyme, then there won't be any formation of your enzyme substrate complex. So, they removes the enzymes and they reduces the formation of your enzyme substrate complex. They will be deactivating or they will be inactivating the enzyme so that they won't be binding to your substrate molecules to form enzyme substrate complex. Moreover, the Vmax value, we have dealt about Vmax, it's nothing but the maximum velocity when your substrate concentration is equal to Km. So, the Vmax value is reduced and inhibition cannot be overcome by increasing the concentration of your substrate molecule. See, just remember that during this suicide inhibition, the Vmax value is going to get reduced. For example, in competitive inhibitions, which comes under your reversible inhibitions, we can revert back the mechanism. You can revert back the effect of your enzyme by increasing the concentration of your substrate. Because of increased concentration of your substrate, your inhibitor will be leaving your enzyme because of the competition between the substrate and the inhibitor for the active site. So, in certain cases with the increase in the substrate concentration, you can go for a normal reaction. So, in this particular suicide inhibition, though you are going to increase the concentration of your substrate, you are not going to regain your original reaction because this inhibitor is affecting the enzyme totally. It is completely inactivating the enzyme. So, as the enzyme is completely inactivated, even though you increase the concentration of the substrate, there won't be any effect on the reaction. So, in this suicide inhibition, Vmax will be definitely reduced and inhibition cannot be overcome by adding extra substrate. In this regard, suicide inhibitions resembles the non-competitive inhibition. Let us see certain examples for this particular suicide inhibition process. So, a common example for this is, you just consider allopurinol. Allopurinol. This is used as anti-gout drug that inhibits the xanthine oxidase activity. This particular component is acting as an inhibitor for this xanthine oxidase enzyme. Because of this, we are utilizing this particular inhibitor as anti-gout drug. Moreover, let us consider that allopurinol is getting converted into oxypurinol. It's nothing but a transition state analog. So, your allopurinol is converted into oxypurinol, which binds very tightly with the MOS, which is nothing but the molybdenum sulfide complex present in the active site of xanthine oxidase. So, I've told you that allopurinol is acting as anti-gout drug. So, how it is acting means, this allopurinol is getting converted into oxypurinol, which will be going and binding to the MOS, which is nothing but molybdenum sulfide complex present in the active site of the enzyme, which is nothing but your xanthine oxidase, because of which this enzyme is getting inactivated. Let us consider another example, which is nothing but your penicillin. We have discussed about the penicillin. It is nothing but your antibiotic, which will be inhibiting the transpeptidase enzyme, which is very much responsible for building the bacterial cell wall, and this penicillin blocks the formation of the cross links between the NAM and NAG. What is NAM? It is nothing but your N-acetyl muramic acid. NAM, and NAG means N-acetyl glucosamines. Generally, in the cell wall of the bacteria, cross links are formed between this NAM and NAG units. Penicillin is completely affecting the formation of your linkages between this NAM and NAG units, and thereby it is creating damage to the bacteria, killing the bacteria. We will just check various compounds that are acting as inhibitors, and the inhibition action of these particular compounds are useful in the therapeutic sector. So, one of such drug is your allopurinol, which we have discussed right now. This allopurinol is getting converted into alloxanthin, and this particular inhibitor is responsible for inactivating the enzyme, which is nothing but your xanthine oxidase, because of which we are using this particular inhibitor or this compound as drug for your gout. Along with this, you can also see another example, which is nothing but your 5-fluorouracil. 5-fluorouracil, which is showing the effect on the enzyme thymidylate synthase. Because of this particular drug is used in treating of cancer. Along with that, we can also take another drug, that is nothing but your aspirin, which is inhibiting the enzyme cyclooxygenase. It is mostly used as anti-inflammatory drug. Moreover, we can also consider DFMO. What is the full form of this DFMO? Difluoromethylornithine. So, this DFMO is inactivating the enzyme ornithine decarboxylase. It is showing the effect on enzyme ornithine decarboxylase, and it is because of which it is useful for treating the sleeping sickness, which we are calling it as trypanosomiasis. So, various compounds are acting as inhibitors. We can consider the positive effect of those inhibitors. We can use them as drugs in order to treat various diseases. This is the positive aspect of your inhibitors. So, coming to the summary, until now in this session we have discussed about what is enzyme inhibition, what are inhibitors, what are the various types of inhibition processes. In that we have discussed about your irreversible inhibition. Under irreversible inhibition process, we have studied about your suicide inhibition process. I want you people to work out these questions so that you can assess yourself how much you have understood from today's class. Moreover, you can refer to these references. This is a textbook and this is a slide share. Thank you.